So the whole thing is regulated by some clock mechanism, but it's not really understood, and that's why, again, I was saying, yeah, with an attractor it tumbles less, but what exactly regulates when it does tumble, and so on and so on. Now, here's where it's going to start getting tricky. During the period of adaptation, now again, remember, that phrase means while the bacteria is responding. Once adaptation occurs, for a model of chemotaxis, again, putting everything together, because you still don't quite know where I was going with this. Let's look at a specific case responding to maltose. It turns out that bacteria like sugar just like we do. So for bacteria, maltose is an attractant. It's going to go after it. Well, one of the many things I left out is that any nutrient, any food supply, any building supply, whatever, it's got to get through the cell wall. It's got to get to the membrane. Well, getting to the membrane, getting to the cell, it's got to go through the cell wall. So there are pores in the cell wall. And these pores are made from porin. Porin is a protein. It's kind of like a, what do you call those portholes in a ship. And you look out at the ocean through the porthole. That's kind of what these pores are, these porin. So the maltose and other food supplies come through the pores, come through the cell wall, to the cell membrane. Binds to maltose binding protein. Maltose binding protein is a receptor molecule. It's very specific. So for example, if we had glucose, we would have glucose binding protein, and so on. Well, this is maltose binding protein, very specific to bind maltose. Now, it turns out maltose binding protein is an allosteric molecule. That's a new word. Allosteric means that this protein is made to change shape. So I've got to back off on something, because earlier this quarter, a couple weeks ago, I was telling that proteins, if you change their shape, you change their function. The shape of a protein determines the function. And if you change the shape, you change the function, and probably deactivate the protein. Well, not in this case. Allosteric molecules are made to change shape. That's their function. No. The light switch, all it does is go up and down. But if you move it, the lights turn on. If you move it again, the lights come on. The light switch itself is still there. But it goes up, it comes down. It's changing shape. That's kind of what an allosteric protein is. Normally, we can say it's in the off position. It's inactive, normally. And then maltose comes along and binds to the protein. Maltose comes along and binds to the protein. That causes the protein to change shape. The wording here is it undergoes a conformational change. Conformation means shape. So, with maltose binding to the protein, binding to the binding protein, the binding protein changes shape. It becomes activated. So I want to repeat this. Again, the maltose binding protein is normally inactive. When maltose binds to the binding protein, the binding protein changes shape, just like I did with that light switch, going up and down, up and down. It changes shape, and it changes shape into the active form. So maltose binding to the binding protein activates the binding protein. By putting it into a different shape, that is the active form. That's what allosteric molecules do. Again, these guys are made to change shape, unlike most proteins. So maltose binding protein is allosteric, will undergo a conformational change, a shape change, in the presence of maltose. So the binding protein now is activated. So what we have is maltose binding protein. It's called a complex. A complex is two molecules bonded to each other. It could be more, I mean, there's no rule. It could be 3 or 4, but in this case 2. So the complex is maltose and maltose binding protein. That complex contains an active binding protein molecule. It's active because maltose is there. Without maltose, binding protein is inactive. The lights are off. So the active binding protein can now bind to MCP. I have there methyl accepting protein, but that's that same methyl chemotactic protein. It does accept the methyl groups. Any questions so far? I know you're all looking kind of groggy and glassy-eyed.
Did it start from this SAM or when it's bonded?
No, it's not there yet.
Okay, okay.
Because there's no methyl group.
Okay.
There's no methyl group. So the SAM is not yet part of it. So this is simply maltose has come through the cell wall, has bound to the membranes, to a receptor molecule. The receptor molecule is maltose binding protein. So maltose binds to the binding protein. Maltose is your stimulus, that's your chemotaxis. So binding protein attaching to the maltose is now active. The active molecule binds to MCP. See, the inactive molecule didn't do anything. It was inactive. So what we have is maltose binding protein, the complex, binding to MCP. That is what sends the signal to the flagella. See, so far the flagella hasn't done anything. But this tells the flagella, stop tumbling so much. We have an attractant. Now, coming back to your question, during the adaptation period, the period of adaptation, while the bacteria is responding to the stimulus, the MCP will become methylated. You know, this stuff. This is going to occur. I need to erase this. So before I erase this, any questions on this before it goes? Okay. MCP is going to become methylated. It turns out the MCP is also allosteric. It's also allosteric. So once the complex binds to the MCP will also change shape. So without the maltose, the binding protein stays inactive. If it's inactive, it does not bind to the MCP. If it doesn't bind to the MCP doesn't do anything either. But the fact is, there is maltose. So the binding protein becomes active, the binding protein binds the MCP, and the MCP, also allosteric, is going to change shape.
Was it already allosteric?
It's always allosteric.
The
I mean, it's just allosteric.
okay.
The only thing is, will it be active or not active?
Right.
It's always allosteric.
Is the MCP that's methylated the one that's connected to maltose and
Well, it is now, yes. Because once the maltose and the binding protein activate each other, it binds to the MCP, so yes.
is the MCP methylated?
Not yet.
Not yet.
Not yet.
Okay.
It's going to become.
Okay.
So when the allosteric molecule MCP changes shape, it exposes glutamyl residues. Glutamyl, that YL, again, it means open bond. The standalone molecule is glutamic acid. That's another amino acid. So you've seen methionine, you've seen glutamic acid. It's another amino acid. And when the glutamic acid is attached to the rest of the protein, it just becomes glutamyl residues. The glutamyl means an open bond. The term residue just means part of a molecule. It's kind of like saying polypeptide. A residue is a piece of a molecule. So glutamyl residues are just pieces of a larger molecule of some sort.
In this case, it's the
What?
In this case, it's the
Right,
MCP.
right. So the MCP, starting off, it's got the usual primary structure, chain of amino acids. It's got the helix, secondary structure, and it's got tertiary structure. You've got your three-dimensional shape. So it's a regular protein. It's got the primary, secondary, tertiary structures. But now the binding protein complex attaches to the MCP. It changes shape. So what happens here is that amino acids that were buried, now they're exposed. That's what the allosteric MCP changing shape does. It exposes amino acids that were buried. So who cares? Well, I don't really want to ask that question actually. But it's these glutamyl residues that actually receive the methyl groups.
That exposed area of.
Yes. The exposed area, this contains glutamyl residues. These are the receptors for the methyl groups. This is where methylation occurs.
Can you say that one more time?
This exposed area is where the glutamyl residues are. This is where the methyl groups go. The glutamyl residues or glutamic acid, either way, I'll take either. These are the receptors for the methyl groups. So methylation occurs here.
So that's the MCP before it changes shape.
Yes?
Then it changes shape, exposes
The residue.
the residue. Of
And then
glutamyl.
methylation can occur. That's why it can only happen in the presence of an attractant. The attractant in this case is maltose. So the attractant will bind to the binding protein, activating the binding protein. The active binding protein attaches to the MCP, causing it to change shape. Methylation can now occur. It cannot occur here because the glutamyl residues are inside. The methyl groups can't get there. It'd be kind of like if you're trying to get a tattoo with your clothes on. You can't do it. And you can't get in there. So methylation will not occur here. So you've got to expose the glutamyl residues. I saw it again somewhere. Oh.
So that MCP is attached to a maltose and a maltose binder protein?
Yes. Yes. That's what triggers and activates this.
Okay, and then that. So.
I didn't hear your
just out of
question.
curiosity, Is it, the methylation occurs at the glutamyl residue, is that because they have the open bond available?
No, well, yes and no. The open bond is not the question is the methylation occurring at the glutamyl residues because they have an open bond? They actually do not have an open bond. The open bond of the glutamyl residues allowed it to attach to the MCP. So as it is right now, there's no open bond. It's just that the glutamyl residues are able to receive the methyl groups. There'll be an additional bonding. Yeah. So again, the methyl group receptor is glutamic acid, glutamyl residues.
Is that also where demethylation occurs?
Sure. So that's where the methyl groups are. So yes.
Does the multi fall off?
Sooner or later. Sooner or later. So let's follow this through now. Remember the period of adaptation and then adaptation? We can now explain that. Because you can add methyl groups, but there are only so many glutamyl residues. There's only so much glutamic acid. Sooner or later they get filled up. You cannot add any more methyl groups. So at that point, adaptation has occurred. You can have all the methyl groups you want, there's nowhere to go. But remember, I said earlier, you can add more stimulus. So let's do it. Add more stimulus. That will make the molecule change shape again. So basically, you're going to unfold it some more. It's going to expose more glutamic acid, which means you can once again get a response. You can get more methylation. The methyl groups will always bind to, well, either way, the glutamyl residues or glutamic acid. I'll take either one. Glutamyl residues or glutamic acid. The more you expose, because of more stimulus, more attractant, the more you expose, the more binding of methyl groups you get, so the more response you get. Do it one more time. Now basically, we've unfolded as far as we can go. All of the glutamyl residues are exposed. So we'll get methylation one more time. Sooner or later, we filled up everything, it's saturated. And now we're done. We can add lots more maltose, it won't matter. There are no more glutamyl residues. So that's when it's really finished.
That was the saturation you mentioned earlier.
Yes,
Yeah.
just exactly. That is what I was referring to earlier. Yeah. So that's why you can add stimulus several times and get a response several times. But sooner or later, you're done. So essentially what it comes down to is, and this is now the answer to the question, how does the flagella know which way to rotate? It depends on which enzyme is being activated. Attractants activate transferase. Transferase is going to add methyl groups. Repellents are going to activate esterase. And the esterase is going to remove the methyl groups.
Causes
Yes?